In this lesson, we're talking specifically about our mandatory prac, growing bacteria and using antimicrobials on them. Yep, let's get this right. Okay, here's our one dot point. It is just the mandatory prac. All right, bacterial growth on agar. Agar is like this jelly-like substance. It's full of nutrients and it's a growth medium for bacteria. Now, the bacteria don't actually eat the agar. They leach the nutrients out of it. Bacteria can grow in broth, right, in a liquid form. They can grow on an agar plate where it's a bit more solid, or they can even grow on a little, strange slope of it. So we'll see different versions of agar around the place. There's lots of different types. There's hundreds of different types of agar and bacterial growth. You will see nutrient agar, right? It's safe to use in a school lab because it's not selectively growing pathogenic organisms. It's just growing anything you put on it, basically. Chocolate agar contains lysed blood cells and it's used for growing really fussy respiratory bacteria. We're not going to use any of that. Blood agar is exactly that. It contains blood from other animals, for example, sheep. Most bacteria will grow on it. There's then different kinds of specific agars, like conky agar, which has some really cute colors depending on what grows. Certain types of bacteria will turn up a certain color and actually change the pH of the agar itself. There's plenty of agar art around, so scientists who participate in competitions every year because it can be quite pretty. The tools we are going to be using for culturing bacteria, however. All right, number one, you need an agar plate. They are going to be pre-made, they are going to be cooled, set, and they are always going to be sterile, so nothing else is growing on them. We are using a Bunsen burner to create a sterile environment because the bacteria won't survive the fire. We are going to use nichrome wire, and this is a wire loop, and it's a delicate tool to transfer bacteria from their culture to your agar. It can be used as kind of like a scooping tool. It kind of looks like something you'd blow bubbles in, or some, Quidditch loop, don't ask me. And they can withstand huge amounts of heat, so they can be thoroughly sterilized between uses. You might use a cotton swab as well. Now, ethanol is used for dipping our tools into before using them to put into the flame. It just helps that sterilization process, which is why you need to keep it away from the flame in case you spill. Aseptic technique is the name given to the procedure used by microbiologists to prevent microbial contamination of themselves or anything else, right? So you can lead to actually growing things you don't want to grow, like pathogenic organisms. Right, it's really, really important to keep your cultures pure. The flame is used to create an aseptic environment. Think of like a 3D bubble around the Bunsen burner, and it eliminates possible contaminants in the area that maybe you're breathing onto, or maybe there's just something else in the area. Now, the tools must be sterilized before use, preferably by dipping into ethanol first and then flaming them. And that's why you're reusing them. Sometimes you can use plastic versions where they aren't reusable, so they're just single use. All right, we are going to do a number of different things, and we're going to make Confluent plates, and also called lawn inoculations. They're used to just cover the agar plate completely in bacteria. And we start off with this as our culture, and we create these plates to get maximum coverage. So the way that I do it is that I will get my loop of bacteria, I will color it in halfway, I'll give it a spin in my hand, I will then color it in here, I will give it a spin again, and I will keep coloring it in this way, and then color it in this way. So basically, as I keep turning it around in my hand, I'm going to get double coverage, and I'm going to go right up to the edges as best I can. And I will show you this in class. We also then create streak plates, and these are used to isolate individual colonies of bacteria on the agar plate. Now, some bacteria grow in like really tiny pinpricks, and it's really hard to isolate if they're all bunched together. And our streak plates, I usually use a 16 streak plate, is where we color in just a little bit of it, and then we flame our loop and clean it off, and then we drag it back through. We don't drag it out of the agar. We flame it again, sterilize it, and we drag it this way again. And eventually, when we get them to this point, we have isolated them. So you should be able to pick up single colonies at that point there. We are going to use antibacterials, and antibacterials are any substance that can kill or inhibit the growth of bacteria, right? And they're going to do it in a number of ways. They might dissolve their lipid membrane, or they might denature their proteins or something like that. When we inhibit growth, we are going to very clearly see it. And the bigger the zone of inhibition, which is what we call it, the zone of inhibition around the antibacterial disc, the better the antibacterial is for that particular bacteria. When we set them up on the plate, we're going to have a lawn inoculation first. We're going to color in that agar plate. We're going to place antibacterial discs on it, and we're probably going to incubate it for 24 to 48 hours, depending on when your classes are, basically. We collect data by actually getting out a ruler and measuring the diameter of the zone of inhibition. Okay, when we collect that data, we can tabulate it, we can do our analysis. We might be looking for differences, and we might have to consider whether things are one-tailed, two-tailed. Okay, this is from a student many years ago of mine whose data I stole. He tested the antibacterial, antiseptic action of various substances like honey, eucalyptus oil, ethanol, things like that. There are a hell of a lot of fiddly technique things we need to be aware of. Hot loops can burn the agar, you can fumble your fingers, you can spill the ethanol near the flame, you can put lids down the wrong way and actually get bacteria on your benches, you can be opening lids up and leaving them open to the world, getting contaminants in, but you need to be prepared before you start. So getting yourself organised before you kick it off is really important. Communicating to your team as well. You've only got two hands. If you're trying to reach over something, it's not going to work. You need to be able to communicate really well in a team. You need to watch your Bunsen burner because you are creating that aseptic zone. You need to make sure you're not putting your arms in it or your bacterial cultures in it. But it will give you a really good understanding of what kinds of things you can grow. But we really don't want to grow anything pathogenic, so we obviously aren't going to open it up, breathe anything in. Okay? Really important that you have a read of your method before you come to class, please.